So someone asked me, and I want to make sure I record it, because I think that if I say it now before the test, you guys might actually pay attention to me. Because I think test anxiety that day of, like anything that comes out of my mouth just goes in one of your ears and out the other. So for the test, you can scroll up and down through the whole test. You can change your answers, although don't change it unless you're absolutely sure. In other words, don't second guess yourself. Right? There's a little icon next to each question. It looks like a little house sideways. What is that? Penta something, five-sided thingy shape. Pentagon, right? 5 sides. Looks like a house on its side though. You can click on it and it'll highlight. And so like if you have a question you're not sure about and you want to come back to, it'll help you find it, right? And then you can unclick it once you've satisfied with it. No timer, right? Although we only have the computer lab for an hour. When you go to the computer lab, which is the Hibernia Center, so go straight there Monday morning. It's in the back of Building 1 next to Einstein's Bagels. It's a computer lab. There'll be an access code that you're given to sign in, to log in on the main computer when you walk in, and then you'll sit down at another computer. You'll click on the lockdown browser software, which the icon looks like this. Right there. On there, it'll take you right to Canvas. You'll log on. So remember, make sure you know your Canvas login, right? Username and password. Some of us have our computers member it for us. You might want to write it down on your phone or something like that in the notes so that you can get in to it. And then you're going to click on the test, and then we're going to come around and we're going to put the access code in for you to be able to take the test. Me and the other person in the room. Once you're done, you're free to leave, log out at the main computer, and off you go. It tends to be a little chilly in that room too, by the way. Just FYI, if you're cold-natured. Any other questions as it relates to the test? I think it's 48 questions.
On Monday, I don't know if I could make it take the test because there's a holiday.
Mm-hmm. Mm-hmm.
So, is there a way I can take it on Wednesday?
You can take it on Tuesday in my office.
What time?
I have office hours from 1 to 3. I have one other person too, so whoever gets the time slot first gets it. So, it's a test.
So, if you want to study, you have to say the objectives.
Study the objectives, right? The PowerPoints go over the objectives, and listen to these recordings again if you need to. And do your homework, right? The last Wiley Plus is up.
You can handle both?
No, that's what I'm saying. I can't handle both of you at the same time, right? So if someone comes at 1, the other person can come at 2. Whoever emails me first gets whatever slot they want. And then I'll say, no, someone else beat you to it. Okay, 1 for you, 2 for you? Okay. Email me so I don't say yes to anyone else. Okay, we're set. Anything else? There's no such thing. We made arrangements. It's over with. No worries. You let me know ahead of time. I appreciate that.
Are you on the first one?
I am in Building 1 in the big cubicle area, the only cubicle with the blue roof towards the back.
Blue roof towards the back.
Yep, you can't miss me.
Is there a number?
38.
38.
Any other test questions? So go straight there. I'll put a sign here in case you forget. Then you'll be late and we'll all laugh at you. Just kidding. All right. So we need a motor though, right? Because we get to finish prokaryotes and do all of eukaryotes, which isn't a lot. And the good news is, those of you guys that had biology somewhat recently, it should be somewhat of a review, right? Eukaryotic cells are us. You study this in biology class. So we'll soon find out if that's true. So where were we? We were just finished endospores, right? And getting ready to start the stuff on the outside of a prokaryotic cell. So we're going to talk about flagella today. Papillae, capsules, and surface arrays known as S layers. So for these, you need to, of course, identify the structure and the function of capsules, flagella. For pili, sometimes scientists designate them. Some scientists just use pili, the name for the sex papillus, for conjugation, where others will also use pili to talk about fimbri, which is a little bit more modified papillae, and that it can be used for attachment to surfaces, and it can even sometimes be used for a form of motility known as twitching. So fimbria, right, refers to plural, and fimbri is singular, if you're just talking about a single one. Papillus, again, just like we know us, is singular. If you're talking about more than one, it's the I, like here. So we're going to look at bacterial flagella, and then later in eukaryotes we'll look at their flagella and how they differ. We're going to look at the structure and talk about how they rotate. And they are arranged in different ways around the cell, and so there's some terminology related to that you need to know. We'll talk about how it is they're able to move and actually know where to swim to, right? How do they recognize things that are good, that are attractants, as opposed to run away from repellents? And then you need to be able to describe the 4 forms. Some of them consider one of the other forms that I'm going to list out. So some books or some scientists will say there's 5 different forms of motility. I just want you to know the 4 major ones, or locomotion. So as always, the animations in your book are fabulous. They're a good, quick, watch.
Vanessa, that last slide you want, you can get that.
You have to be in presentation mode for you to click on it, and you have to be on the internet. Notice it says you must be connected to the internet in order for it to play. It basically just links to your book. So motility, for the most part, for bacteria is via flagella. So the flagella itself is a spiral grouping of proteins. It's actually a hollow tube. It is rigid, so it's not flexible. And the location of how many flagella they have and where they're located on the cell varies depending on the organism, on the species. So this spirillum, right, this spiral-shaped bacteria, notice it has 1 flagella at either end. And remember, we wouldn't be able to normally see this with a light microscope. You'd have to do a special flagella stain, right, that they did for this light microscope picture, so to make these actually thicker than they are so you could see them. This, on the other hand, is a transmission electron micrograph of Salmonella, and they colored it. Remember, this would normally be a black and white picture, but they kind of colored it to accentuate the different components. And you can see he has multiple flagella all around. So the term we use for that is peritrichous. Trichous means hair-like, peri meaning many. And E. coli looks like this too. He's just covered in flagella. So here's probably another scanning electron micrograph, and you can see it's just covered in flagella. What shape is this bacteria? Rod-shaped, or the term is bacillus, right? But the term for how many flagella it has and how they're arranged, the fact that he's covered in it, almost like the scary cousin It from the Addams Family, right? It's peritrichous. When they have a single flagella, like you see here, that would be mono. Mono, of course, meaning 1, trichous. Sometimes it's also referred to as polar, because notice it's at the end, right? Polar meaning at the end. This next one, what shape do we see here? Spirillum, right? Both of these pictures are spirillum. For this one, notice there are multiple flagella at both ends. This term, lophotrichous, refers to actually just a grouping a flagella at an end. And in this case, this guy has it at both ends. But if he had it just at one end, it would still be the same term for the flagella arrangement.
What's that term mean?
Lophotrichous means a clustering of multiple ones, multiple flagella at one end. Or, in this case, both ends. Now for this one, there's one at each end, right? You see that? One flagella at each end. This is known as amphitrichous. Anyone know what amphi, that prefix, typically means? Opposite. So there's one at each opposite end. So these guys, their flagella actually spin. And you don't see it in this picture, you'll see it in another picture that I'll show you guys later. It has kind of a hook at the end. So they spin around like a propeller does on a boat. And what propels this motion, this spinning, this rotating of the flagella for bacteria, is proton motive force, where they're pumping protons across their membrane. And then a pore opens and the protons come flowing back through. That actually spins the basal body embedded within the membrane and the cell walls. So when they turn it a particular way, just like when you're driving a boat, right? When the propeller goes a particular way, you move forward, right? If it moves the other way, you move backwards, right? For them, when they move their propeller a different way, they tumble. So that's how they change direction. And they don't do it very well. They literally just tumble around. And then they start running again. Now, how is it that they know they want to go in a particular direction? Anyone know? Or run away from something? They don't have eyes and ears like we do, right? Or the sense of smell even. They have protein receptors in their membranes, right? Sense the environment. So things attach to those, right? And if it's something good like food, they're going to keep heading towards that direction, right? That signal is going to be sent inward to that cell. And they're going to run more than they're going to tumble, right? As long as they're still heading towards what it is they want. Or heading away from something like a repellent. When there isn't any stimulus to get them to go in a particular direction, they'll tumble around more than they'll run. Right, so he's just tumbling around here, right, mostly running a little bit. Here, he senses something he wants, it's an attractant, and so you see he has much longer runs than when he tumbles. And the same thing for a repellent. When you want to get away from something that's bad, right, you're going to run, obviously, as much and fast as you can, and not tumble so much. So yeah, it's chemoreceptors, right, that sense the chemicals in their environment, and they are proteins embedded in their membranes. Not all cells have external flagella. We looked at one cell shape that actually has external flagella that are within an outer sheath. These are referred to as axial filaments. Anyone remember which ones have this that move like a corkscrew? And they're helical in shape. Can cause Lyme disease. Spirochetes. Spirochetes. Some spirochetes have flagella in the periplasm. So what about if they don't have flagella? Does that mean they don't move? No. There are some of them that can actually glide, especially on solid surfaces. This isn't well understood, this type of motility. Some scientists believe that they're actually produced an extracellular slime that helps them glide. And twitching motility is relatively new where they're using the papillae, these protein appendages, where they can attach and kind of creep along surfaces. Now, the sometimes accepted fifth possibility of motility is where some organisms will actually get into our cells and use part of our cytoskeleton, the actin or microfilaments, they can attach to, and they can activate them and use them to shoot themselves from one cell to another. So Shigella does this, Listeria does this. So they're only motile, though, when they're using our machinery. Right? They're using our proteins, they're attaching to them, and they're using them to propel them from one cell to another. So that some people consider this a fifth type of motility. So what are the four forms to review? The most obvious is if they have what? A flagella, right? And how does that move? It rotates. And then the next one was our specially shaped ones, right? With modified flagella known as axial filaments. And these are our spirochetes, right? So the corkscrew motion of the spirochete due to their periplasmic flagella known as axial filaments. And then the next two we just named, gliding and twitching, using papillae commonly referred to as fimbriae. And then the last would be polarization of actin within our cells. So this is really important for pathogenic bacteria. A lot of them have papillae or papillus, and specifically ones that we call fimbriae or fimbria, because this allows them to adhere to us. So to give you a specific example, Neisseria gonorrhea, the bacteria that causes the infection we know as gonorrhea, it is able to attach to our epithelial cells because of the fimbriae it has, these specialized papillae. So it'd be really great if we could make a vaccine against that, right? You know, make antibodies that'll bind to it before it binds to us. Can't do it, because they're constantly changing the proteins that make up the papillae. So you can get this infection more than once, can't you? You don't become immune to it, because it changes. What's the good news with gonorrhea, though? You know how you get it, right? You can kill it with antibiotics, right? It's a bacteria. The bad news with that, y'all, is the misuse of antibiotics. There are some strains that are resistant to antibiotics. But it's a sexually transmitted disease, right? So you could avoid getting it if you choose very wisely who it is you play with and how you play with them. But yeah, it's specially designed so it can stick to us. And that's the first step for any type of pathogen. They have to be able to adhere to us. They have to be able to grab on. So in addition, some of them, the papillus that they produce, you can see in this picture, you see this tube? It's a protein tube, the papillus, the sex papillus, and what happens is genetic information can travel in between these bacteria. Typically it travels from one to the other, not back and forth. So you can see why it's referred to as sex, because that's what sex is, y'all. It's an exchange of genetic information. All little spermy guys are mobile DNA. So in a sense, you know, bacteria don't have to sexually reproduce, but they can share genetic information, what we would consider a sexual act. But they do this through that protective protein tube that they'll use to attach to each other. And then you can see both of these pictures here, right? All these little lines. That's papillae, and if they're using it to stick, right, or adhere to surfaces, it's sometimes referred to as fimbriae or fimbria.
So the one on the right, just to go back, the one on the right is actually connected to the other?
Yeah, these two are connected to each other via that long red protein tube. And DNA is going from one cell to the other through that tube. Yeah, the papillae that's typically used for attachment, and they'll have a lot of them. Like, I mean, look at this guy's like covered. And these are much finer than even flagella. And again, they've seen that some of them actually can use this for a form of motility where they kind of creep along, they kind of twitch along. So another outer layer that some microorganisms will have, and I say some microorganisms because this isn't just unique to bacteria, though it's very common in bacteria. But some eukaryotes, like yeast, produce a capsule layer. And typically it's made of polysaccharides, so many different sugars. And it's usually pretty uniform. So this is a special stain, a special capsule stain that you guys in the lab will do next week, where you can see the bacteria has been stained and the background's been stained, but this big clear halo around is the capsule. So this is outside of the membrane, the cell wall, and if it's a gram-negative, even outside the outer membrane. And why it's really beneficial and why a lot of pathogens have it is it helps them adhere to us. Right? Because remember, that's the first step of infection. They have to be able to stick to adhere. And this additional layer helps them. It also is almost like they're wearing a disguise. When they get into our body, it's really hard for our body to recognize them. Our phagocytic cells that would normally gobble it up and eat it have a hard time recognizing it. Unless we have produced specific antibodies, maybe from a vaccine or a previous exposure, it usually takes us a while to detect these guys. And so they're growing and destroying and eating us.
That's their defense.
Yeah, it's one of their defense mechanisms. Well, just to have that capsule layer, right? It just have that extra layer that helps disguise them, and it also helps them from drying out in the environment. And capsules can help them, one, it helps them stick, and then they'll produce even more sugars and even more matrices in complex with other organisms nearby and create a layer that we know as a biofilm. The bad news with biofilms is that they're really hard to penetrate, and what plaque on your teeth is a biofilm, right? It's bacteria that are stuck to your teeth, to that non-shedding surface. So what do you have to do to remove them? You physically have to scrub with a toothbrush, right? Or floss. You can't just rinse your mouth out. You're not going to get them to go away. Because they are physically stuck to that surface. So when you get at home, if you haven't cleaned the tub in a while or the sink, right, you get that scum and you got to scrub, right? Or you got to use the scrubbing bubbles. You got to get something that's going to get in there in between and break up these connections that these organisms form with each other. So here's some, pictures, and, it forms even on your toothbrush. So the last is something relatively new that's been discovered too, is these protein, almost like, I don't even know how to describe it. It's like an armor, right? Does it even look like armor? So this is again, this is the outermost covering of some bacteria found in both gram-positive and gram-negative. Oh, look, they even use the word armor. Huh. But I mean, it looks like that, right? And the really cool thing about this protein coat known as an S layer is that viruses that attack bacteria can't attach to this surface. And so the same thing, like they can't get infected because the virus can't attach. And the same is true for us with viruses, right? Viruses have to first attach to the cell before they can gain entry. So if you can have something that stops them from attaching, then it helps protect you. So it makes sense that, the ones that have evolved this layer to have another layer of protection would be beneficial. So that's it for prokaryotes. Yay! So last is eukaryotes, and I had given you guys a little homework assignment, right? Because one of our objectives is to compare and contrast prokaryotes to eukaryotes. So let's see how you did. So remember, within the organization of life, we have 3 domains that we talk about. Bacteria are what cell type? Prokaryotes. And archaea? Prokaryotes. And that's why in that previous lecture we talked a little bit about archaea as well, right? And specifically the differences they have as it relates to bacteria. And then this whole lecture is going to be on eukaryotes, which have their own domain known as eukarya. But that's the cell type, right? Prokaryote versus eukaryote. And of course the defining characteristic of this cell type is what? The nucleus. And which one has it? Just eukaryotes. Who has DNA that's complexed around proteins we know as histones? Right, eukaryotes and archaea. So you have these similarities between these 2 different cell types right here. And yet differences within the ones that are the same cell type, right? The nucleolus is something new for today, so we know it's in eukaryotes, right? But what is it? What's the nucleolus? It's not a misspelling of the nucleus, by the way. It's a region inside the nucleus where something happens. Anyone know what? Close.
It's inside the nucleus?
Inside the nucleus. So definitely transcription, but transcription of something that we need a lot of. What do we make? What makes proteins? Ribosomal RNA, right? Lots of ribosomal RNA to make what? Ribosomes. So lots of ribosomal RNA to make ribosomes. That leaves the nucleus and complexes with other protein to make the actual ribosome, which then makes proteins. All cells have ribosomes, but remember, only eukaryotes have that defining wall, double membrane around their DNA that forms the nucleus. So because of that, all the ribosomal RNA tends to form in this one region where the DNA code is for making the ribosomal RNA. Because, it's just continually being made. Because you need a lot of ribosomes. But then it leaves the nucleus to complex with the protein to actually form the ribosome. So mitochondria and chloroplasts are sometimes referred to as organelles, right? So this level of organization. Who has them? Just eukaryotes, right? And of course not all eukaryotes, right? Only photosynthetic ones have chloroplasts. And of course there's an exception to mitochondria. There is one group of eukaryotes that don't have mitochondria. But for the most part, everybody, even plants, have mitochondria. And in addition, chloroplasts. So we talked about plasma membrane lipids, right? And so whose lipids were really different here? archaea, right? Because of the living in the extreme environments. And what was one unique thing that those lipids could do that you don't see in bacteria and eukarya? So when we say a membrane, how would you describe a membrane to me? It's a protective covering. Be more specific. What's it made out of? It's made out of lipids, proteins. How is it arranged? So you have those lipids, but they're not just lipids, they're what? Yeah, heads and tails. What do we call the heads? Phosphate groups, right? Yeah. So we have phospholipids. How are they arranged to form the membrane, though? Tails towards the inside, but you said tails. You put an S on that. There's more than one tail? Bilayer, right? Normally it's a bilayer. What can some archaea do? It's all one, right? They form monolayers. So remember, the linkage between that phosphate and the fatty acids is ester, but the important thing here, right? Remember I said I wasn't going to make you memorize the chemical name? The important thing is that eukarya and bacteria are the same. Archaea is different, right? It has that different linkage, it has branched fatty acids, and it can form monolayers. And again, these are our extremophiles. These are the ones that live in those extreme environments. So it makes sense that their membranes are quite different. Although they function in the same way, they're semi-permeable barrier, right? But they have to have these additional modifications to survive in those extreme environments. So the flagella for bacteria and also archaea, we just learned, rotates, right? Does anybody know how the flagella, and I should have put movement there, sorry I didn't. Anyone know how eukarya, how their flagella move? They wave, right? Yeah, they wave. They don't spin. They wave back and forth. And they do that because of part of their cytoskeleton that makes up the flagella, known as the microtubules. So what's the cell wall of bacteria made out of? Peptidoglycan. What's the cell wall of archaea made of? Not one we use very often, right?
I found it somewhere that says varies from peptidoglycan to protein.
Well, it's commonly referred to as pseudomorine because it's similar to, right,
Is that like the same?
peptidoglycan, but one of the sugars is switched out and they have the different form of the amino acids. And some of them actually are made out of protein too, you're right in that. What about eukarya? First of all, do we have cell walls? No. So it varies, right, among the different groups. Plants do, and you know what their cell wall is made out of? Cellulose. In fungi, theirs is made out of the same thing insects' cytoskeletons is made out of. Chitin. So we're going to talk today about the size of the ribosomes. Right? It's referred to as the Svinberg system of measuring. It takes into account the weight, but also you can say the aerodynamics of that molecule as it moves through a solution. So the important numbers that you will see in the literature is the size of both bacteria and archaea is 70S, where eukaryotes are different. Ours are 80S. Is there a cytoskeleton in bacteria and archaea? Yeah. Is it as complex as eukaryotes? No. Right, but it's definitely there. It's relatively simple, but it's there. For eukaryotes, what are the major components that make up the cytoskeleton in cells? We've already named one of them. Microtubules, right? What else is there? They all have micro in the name. Microfilaments, intermediate filaments. Oh, so they don't all have micro. And then microtubules. So we'll look at those specifically today. And then I added this last block, so don't lose your mind, right? It wasn't in yours. I forgot it. Does anybody know what plasmids are? So what are they made out of? What molecule? They're DNA. And they're very common in both bacteria and archaea. And some eukaryotes they've found plasmids in. These are extra chromosomal information. So in addition to the chromosome, they have these additional circular pieces of DNA. And they don't have to have it. They can survive without it. But it can be beneficial, and it can be really bad news for us, because a lot of those plasmids out there have things like beta-lactamase. Why don't we like beta-lactamase? What is that? Bacteria's protection against... It's an enzyme. It's an enzyme. Beta-lactamase is an enzyme. It protects them against what? Antibiotics, right? Like penicillin that have the beta-lactam ring. That enzyme breaks it. Look, imagine this. A bacteria has that information on how to make that enzyme on a plasmid. So he's resistant to penicillin and all beta-lactam drugs. Then he comes along, one of his friends, who doesn't have that information. So they're like, oh, let's conjugate. Hooks up the little sex papillus, and he gives that plasmid to his friend. His friend now has beta-lactamase. He can resist beta-lactam drugs. So that plasmid is actually referred to as an R plasmid or a resistance plasmid because it actually contains genetic information to be resistant to antibiotics. Kind of scary stuff. So as with prokaryotes, for eukaryotes, if I give you a drawing or a picture, you need to be able to identify the major components that we'll talk about. So again, hopefully a lot of these organelles are review, right, from biology class. So the membrane, not all that different from bacteria, but we know archaea have quite interesting membranes. But the dividing characteristic is this additional membranes inside the cell organized in a way that we've compartmentalized things, right? We've organized things at a smaller level. So that's what organ means, right? It's an organization. L's refers to the fact that they're small. So the nucleus, we know, is that defining characteristic, in that region within the nucleus of that really densely packed ribosomal RNA is referred to as the nucleolus. And as we said in our intro, some eukaryotes do have a cell wall. Plants, algae have cellulose cell walls. Fungi have chitin. Diatoms actually have silica, which is what glass is made out of. It's kind of cool. So we need to know the main function, right, of each of our organelles. The nucleus, of course, is the command center. It's where all the DNA is and where it's transcribed. right, where the RNA forms. But then it has to travel out of the nucleus into the cytoplasm to be translated into proteins. The mitochondria is where the energy production happens for most cells. If they also have a chloroplast, they can do photosynthesis. They have a rough ER, and what makes it rough? Anyone know? The ribosomes that are attached to it. So this is for protein. The Golgi apparatus is a stack of membranes that modifies proteins and transports them where they need to go. Vesicles are, again, membrane-bound storage. So what was similar, what did we call storage compartments within prokaryotes? They weren't surrounded by a membrane, but they stored stuff. What do we call them? Inclusion bodies. Inclusion bodies. Lysosomes and peroxisomes, these are specialized vesicles, so membrane-bound. Lysosomes are really important, especially for our white blood cells, because they contain digestive enzymes. So remember, we talked about an enzyme called lysozyme, right? That destroys peptidoglycan. So that's the job of these vesicles, is destroy things. Same thing with peroxisomes, right? They break down fatty acids. And again, we do these things within membranes so that we can control the situation, right? We don't want these enzymes just running free in our cytoplasm. They would chew up anything they came across. We want to be specific on what they're digesting, right? So we stick it in the vesicle, surrounded by a membrane so it can't get out. Hydrosomes, they believe, these are a remnant of the mitochondria for that group of organisms that don't have an actual mitochondria. So they do some type of energy production, but they use hydrogen to do it. That's why they're called hydrosomes. But again, membrane encased. So we already said about the nucleolus. Let's go over that. So for the ribosomes are made out of protein and ribosomal RNA. And they are the site where proteins are made. So they attach to the messenger RNA and read it and make the proteins. For prokaryotes and eukaryotes, the size differs. So we've already said the overall size of the two subunits together is 70S for prokaryotes. It's 80S for eukaryotes. And remember, this was actually how we ended up with the 3 domains. Because even though the size is the same for bacteria and archaea, their sequence, their RNA, ribosomal RNA sequence is dramatically different. Even the amino acid sequence is different between bacteria, archaea, and eukaryotes. And it was these fundamental differences that gave us the 3 domains that Carl Woese came up with from his research of looking specifically at the code. And of course, all cells have to have these huge protein RNA structures in order to make proteins. And so all cells have this. So the 2 different subunits, as you see, if you were to add them together, it doesn't come to the total we're used to, because remember I said this measuring system isn't just weight. It's also aerodynamics. So you notice the numbers do get bigger when you put them together, but it's not the simple math that we're used to, right? Of 40 and 60 equaling 100. Instead here it's 80. Because this system of measuring takes into account 2 variables, weight and aerodynamics of the molecule. You guys just need to remember the whole size of the whole ribosome, 70S versus 80S. So how do things know where to go within a cell? So if a protein gets made in the endoplasmic reticulum, how does it know, say, it needs to go outside of the cell, or into a lysosome, or travel to the mitochondria? How do the proteins know where to go? right? It's programmed in the RNA, and there's a special sequence on the protein itself that the cell knows where the stuff needs to go. And because the cells are so much more complex, right, than prokaryotes, we actually even process our messenger RNA. And we process our proteins a lot more than prokaryotes do. So there's a lot known. Obviously, I don't expect you guys to know the details here, right? But just know that stuff is coded, basically, right, as to where it's going to be transported to. And that's part of the job of some of these organelles. Of course, the rough endoplasmic reticulum has those ribosomes associated with it, so it's making proteins, right? But it's passing them into travel vesicles or transfer vesicles that will travel to the Golgi apparatus. And other components may be made in the smooth ER, and those are brought to the Golgi apparatus. And so my nickname for the Golgi apparatus is the packing plant, right? It packages everything together and sends it off to where it needs to go, whether it be outside the cell or to some other area within the cell. But all that stuff is coded in the proteins themselves. So there are some specialized pathways that happen within the cell. That's why they're called endocytotic pathways. And of course, it's important to have the building blocks. In this ability of doing phagocytosis or endocytosis or autophagy, we need the ERs, right? We need the smooth ER, we need the rough ER to make the building components. They go to the packing plant, and for endocytotic pathways, we need lysosomes. We need those vesicles with the digestive enzymes in them. Then the cell can bring stuff in and surround it in a membrane by just regular endocytosis, but it's going to fuse that vesicle with a lysosome. And again, as I said, we're going to digest within the lysosome under safe conditions encased in that membrane. Or some specialized cells, like our phagocytic white blood cells, they're called that because they do the process of phagocytosis, where they take things in like bacteria into a vesicle. They surround it by a membrane. They call that membrane a phagosome, that vesicle. Then they're going to fuse it with a lysosome, and guess what they do with the name? They fuse it. They call it a phagolysosome. But what are we doing? We're digesting that bacteria. We're literally eating it. Mitochondria and chloroplasts can reproduce on their own. There is the belief that they were once free-living prokaryotes all on their own, and they were engulfed. And we began this symbiotic relationship. Anyone know what evidence we have that they were an organism all on their own? What do they have that's just theirs? They have all these membranes and such. They have their own DNA. They have their own ribosomes, and guess what they are? 70S ribosomes. But they don't produce all their own proteins. Some of the proteins they need are actually produced by information that's encoded in the nucleus. So it's going to be transported to either the chloroplast or the mitochondria. But when the cell needs to recycle them, they engulf them themselves into a vesicle, a lysosome, and that's called an autophagosome, and that process is referred to as autophagy. And I'm out of time, aren't I? So make sure you review the rest, because it's on the test. Yep. Make sure you know microtubules, microfilaments, right? I'll try and post a clip of an old lecture to help you guys out too.
You still like to send me an email on the website?
Yes. So I'll put you in my calendar. And like I said, I won't let someone else come. Yes.